First of all, I really would like to thank you for coming here this evening. My name is Ken Stewart. I'm the president of Seattle Biomedical Research Institute, or Seattle BioMed as we call it. And we're really happy to see you here tonight to have a discussion about malaria vaccines. So we have a very important mission at the Institute. We're striving to eliminate infectious diseases that are killing millions of people every year. And that statement always gives me pause, not so much because that's a big number, but because that's the tip of the iceberg of the problem. Because for every person that dies, there are many, many more who are suffering disease and disability. And then there are economic and other consequences of these diseases. So these are huge problems. So we are working every day to develop the vaccines and drugs and diagnostics that are going to be part of the solutions for the control and elimination and hopefully eradication of these diseases. And the diseases that we work on are HIV, AIDS, TB, malaria, and the green diseases we call neglected tropical diseases. This evening, we're really pleased to have our friends and collaborators here from PATH. We used to share a building with PATH on the ship canal. We used to joke that we were working at opposite ends of the spectrum of work that needed to be done, but we were on a convergent course. And in fact, we have, I wouldn't say converged, but combined. And now I can say that we're really pleased to have PATH back in proximity and neighbors just within, not my throwing distance, maybe my son's throwing distance, down just a block away. And actually, I had the pleasure last night of bumping into Chris Elias in the Starbucks across the street. And that sort of interaction, that immediacy of interaction, we really want to put to work. So I'll give you a brief overview of what we're going to do this evening. It's been a little bit, it'll be a little bit different from what we've done before. So how many of you have been here to Global Health 101 before? Please raise your hands if you've been here. All right, about 10%. That's good. So welcome back. And for the rest of you, I hope this will be a great learning experience. We used to have what we called speed dating, where we'd break up into about 4 groups and give people time to actually sit with the investigators, hear about the work that they're doing, and most importantly, to be able to ask questions and hear their answers. And so there will be a question and answer period tonight with the panel. And I would encourage you to ask questions, and I like to say, especially the people in my lab, the only question that's a dumb question is the one you don't ask. Because if you don't ask a question, then you won't learn something new, and it's okay to be wrong. So vaccines are going to be a key tool in the armamentarium to eradicate the infectious diseases that we care about. And tonight, we're going to focus on malaria, which kills over nearly 1 million people each year, mostly children under the age of 5. And so I'd like to introduce, first of all, Dr. Regina Rabinovich, who we call Gina, by the way, who will lead the panel discussion. So I'll give you a little bit of background on Gina. She is the director of infectious diseases in the Global Health Program at Bill and Melinda Gates Foundation. So in this capacity, she oversees the programs for drug and vaccine research and development, as well as clinical trials and introduction and control programs for the prevention, treatment, and control of global infectious diseases. Diseases including HIV/AIDS, TB, malaria, pneumonia, diarrhea, and neglected diseases. That is an awesome responsibility. Just amazing, frankly. Makes our mission seem very, very focused. Regina has extensive training and experience in many aspects of medicine, including maternal and child health, epidemiology, and preventive medicine. She received her M.D. from Southern Illinois University and her Master's of Public Health from the University of North Carolina at Chapel Hill. She was at the National Institute for Allergy and Infectious Diseases for a time. I'm sure that's resonating in her head as I say for a time. And she became chief of the clinical and regulatory affairs branch of the Division of Microbial and Infectious Diseases. She became the first director of the Malaria Vaccine Initiative, which is where I first met her. She then moved to the Bill and Melinda Gates Foundation, where she shouldered the huge responsibility that I just described. If that's not enough, Gina also serves on boards, including those at NIAID, the Global Fund of HIV/AIDS, Tuberculosis, and Malaria, Medicines for Malaria Venture, and the Institute for One World Health. As you can see, she's a true global health champion. And I know she's doing more than that. That's just some of the things that she's involved in. I think it's particularly fitting that Gina moderate this panel here today, because our first contact was around the start of the malaria program that was here. That was a little over 9 years ago that contact happened. And she was at MBI, is now, was at MBI, and now is here at a time when we are beginning to see the first fruits, hopefully, of the investment of her time and effort, as well as the funds from the foundation and others for a vaccine that is now beginning to be tested in man. So all of this is really a testament to her vision, partnership, and hard work. Gina.
It's actually great to be here, and I've never chaired a panel that is more like malaria vaccine dating, but I like the concept. I'm going to hold myself to 5 minutes, and I'm going to hold each of our speakers to 5 minutes, and then I'll start giving them dirty looks, and they'll know that it's time to move on. Because what I want to do is open it up to be able to see what questions you have and to generate the discussion that I think could be a lot of fun. Now, for any of you that kind of wandered in here, because there was drinks and dinner, et cetera, and you don't know what malaria is, that's okay too. We'd love to talk to you more about this. Malaria is a parasitic disease. And by that, I mean something that's transmitted to humans and courses through the bloodstream and is transmitted to humans through a mosquito bite. And the vector that infects us sits around the United States, but it's also at much higher intensity and efficiency in places like Africa. You spoke about 1 million deaths. I don't know, is it 800,000? Is it 1 million? That's way too many. And the number of cases that generate those 1 million deaths is what really gets overwhelming when you think about who needs a vaccine or who needs treatment. It's probably 250 million cases a year even now with the massive effort to distribute bed nets. And so that means that reflects the tip of the iceberg. It reflects a population at risk that's approximately 40% of the world's population. So that's why malaria is important and why these people have committed their careers to the part that they're going to tell you the story about. The major approach to malaria, now since let me tell you the truth, we don't have a vaccine. Wish we had a vaccine, that's what their job is. So what can you do out in the field right now? Well, if you travel to Africa, you will spray your clothes with insecticides. You will sleep under an insecticide-treated bed net. You will take, because you can afford, you can take drugs that will actually basically immediately treat you. And so you can prevent getting the disease. Unless you're really unlucky and you do all those things and you still get it like I did. I can tell you it's a non-fun disease. My husband who's in the room knows I came to him and said, can you please take me to the hospital? And he said, well, why do I need to take you to the hospital? You travel all over the world. And I said, because I can't drive. And I showed up at the hospital with a temperature of 105, which is typical for an immune-naive person. The road towards a vaccine has been long, and there's been a lot of blind alleys. There's been a lot of 5-year initiatives in Australia, even in the United States. There's been malaria eradication efforts that have failed. And so the only lesson I can tell you of that is to beware of the 5-year plan. This really takes a longer vision for impact and to be able to affect change. Now the good news is we're in the midst of a new era. And I think that era is reflected by malaria, but not only by malaria. It's really part of what I'm calling the global health revolution, akin to maybe the green revolution that took place in agriculture once upon a time. You're going to hear more about a vaccine that is now in phase 3 trials, and what a phase 3 trial really means. It's very astonishing and complex, and I'm thrilled to be watching this unfold and waiting for the data. I work for people who are impatient, and they also want to see the data. There's totally new types of approaches to vaccines. The whole genetically attenuated and irradiated attenuated parasite type of vaccines, and you're going to hear about that. And all of this has happened at a time when the concept of eradication has been raised and started with discussions that took place in Seattle. Because the concept is you can prevent this disease, for most people other than me, with bed meds and with pre-treatment. You can actually treat this disease. We can afford it. And now ACTs are more and more affordable and are being funded by these big global groups. So it's treatable, it's preventable. We can create programs to manage it, and at what cost? It became very clear when the cost is a significant amount and percentage of ODA, overseas development assistance dollars, is that even with effective drugs, even with effective vector control, that we're going to need a long-term strategy that the world can afford to sustain. And if you can't sustain it at that level, then you very much better figure out an end game. And so the question was eradication. I think the challenge for us today is that vaccines can, I'm not even sure will, this is what the data will prove, play a particular role in the whole eradication concept. And it all depends on how good they are and where they work and when they work. And so the research and innovation that underlies the progress in malaria really is reflected by two things. One is the progress in vaccines, and the second is, which I hope you'll hear from the speakers, the partnerships that it took to get us here. Now we have 4 great people, and so let me go ahead and just introduce all 4 of them so we can get that part out of the way, and then they're going to introduce each other as they speak. The first is Dr. Christian Loucq, who is the director of the PATH Malaria Vaccine Initiative. I'm happy to say all 3 living directors of the MBI are in this room today, with Melinda in the audience. And MBI seeks to accelerate the development of promising malaria vaccines and ensure their availability and accessibility to developing countries. He has more than 30 years of experience in pharmaceuticals, in global health, in medicine, and in vaccines, and has worked for large companies, and we're delighted that he came over to the Malaria Vaccine Initiative. Dr. Ashley Birkett is next to him, and he directs the identification of promising malaria vaccine technologies and implementation of partnerships with both for-profits and not-for-profits. He has 15 years of experience in vaccine development and has worked primarily with biotech companies, which is where I first got to know him a decade ago. And prior to joining PATH MBI in 2008, he was Senior Director of Preclinical Research at Campus, a biotech company. Our third speaker is Dr. Stefan Kappe. And Dr. Stefan Kappe has joined Seattle BioMeds, and I guess you weren't here when I first came to visit, the malaria program in the fall of 2003 as a principal investigator. And the focus of his research has been pre-erythrocytic stages of malaria in the human host, and he is in the process of developing a genetically attenuated malaria vaccine that will go into trials really soon. The project is funded as one of the grand challenges in global health projects that is part of a program funded by the Bill and Melinda Gates Foundation, and we're delighted that he's here and arrived from New York University, where he was an assistant professor working on blood and liver state malaria. Then Dr. Angela I said Talley, am I saying it correctly? Is the lead trial investigator for the Malaria Clinical Trials Unit here at Seattle BioMed. She's been here since 2008, and she's active faculty also at the University of Washington Medical School in the Division of Allergy and Infectious Diseases, as well as the Department of Global Health. And so she's recently participated in trials in Tanzania, worked in fields like maternal vaccination of pregnant women, and establishing the protocol challenge model for vaccine trials that I think will be implemented at Seattle BioMed. So without further ado, let me go ahead and ask Christian to please come here, because I understand they're using something called PowerPoint.
So here we are, a group of people. We've got the Malaria Vaccine Initiative today, the PATH Malaria Vaccine Initiative is the global program of PATH. We're a group of 50 people today. 50 people mainly, our main location at the moment is Bethesda in Maryland. Why? Because we are working a lot, there's a lot of research conducted there on malaria. I can explain a little bit the history of our location. And we're a group of people, it's a group of people made of comprising guys coming from industry, scientists, and people with a global health background. And here we are, we don't have labs, we don't have a plant, but we have as a mission to manage partnership and fund to select promising candidates and to look into accelerating all the promises that we see in labs and in industry. Because one of the major problems of the development of the malaria vaccine today is that there is no market. The malaria vaccine is made for endemic country, and malaria is endemic in sub-Saharan Africa. And unfortunately, there is not enough market to justify the investment to develop such a vaccine. As I said, we are a global program of PATH, like other vaccine programs. And how do we work? Means in fact, and how I've been working for the past now 11 years, and the past 3 or 4 years that I've been there already. We partner with industry, we partner with large pharma, biotech, we partner with government services, we partner in fact with academia, and in those centers we select programs that we are going to invest, which we are going to define with those centers milestones deliverable on an industrial model to bring those programs out of the lab and as quickly as possible along a process that is going to go to the various phases of development of a malaria vaccine. You're going to tell me why have been working such a long time on malaria vaccine and we don't have a vaccine yet? It's a parasite. Parasite is a complex thing, and there is not yet a vaccine developed against such a bug. So this one, if it is the first one to reach, in fact, market, is going to be the first parasite vaccine against a parasite. As I said, there is unfortunately no market, little interest and little pool, to attract investment and push industrialists to put more money. So if we don't share the risk, we're not going to see a vaccine coming to market and being developed. Going to, maybe, just to tell you what Gina said, we have today tools. There's a lot of money which is used today for malaria, but we see already, and unfortunately we do see with the best of the drugs that we have, beginning of resistance coming up in Southeast Asia. The insecticide that we are using on bed nets have been around for several decades. There could be a problem there. And we all know that if we want to finish the job, we will need a vaccine to complete the job. We say we partner, and that's a list of all various partners with which we are working. And you see Seattle BioMed among those partners. We are very proud of working with this organization, and we are going to discuss a little bit further this evening what we are doing. Most important, and that's the excitement of the day, we are now with RTSS in phase 3. What does that mean? We are in 7 countries, 11 centers, enrolling, and as I'm speaking this evening, we have already close to 9,000 kids of 5 to 17 months of age enrolled that have received their doses of vaccine, and we have close to a little bit more than 1,000 infants that are going in the EPI, the program of vaccination, that have also received the vaccine. It's a major undertaking. 16,000 kids are going to be immunized and followed for the next 30 months across 11 centers. our pride, 11 centers with scientists, African scientists, that are working, doing top-level science in developing a malaria vaccine and in driving that phase 3. And not taking much more time, because I think that I'm going to come to the end of my 5 minutes. What is that picture? That picture is a picture of the 5 first kids that were immunized. And it was in Ifakara in Tanzania in May, I think, April, May, I think, of 2009. And that's a little bit the last partnership that I want to highlight. 16,000 kids means 16,000 mother and father that are partnering with us because they are trusting the development that we are going, and they are bringing their kids to do the trial and to contribute to what we are developing at the moment. Thank you.
Thank you. And just to move on from where Christian started, tremendous achievement in moving the RTSS vaccine into phase 3 clinical trials in Africa. But that's not the end of our work. That vaccine has tremendous promise, going to save hundreds of thousands of lives in Africa if it's finally approved and administered in vaccine programs in Africa. But we need better vaccines that are even more effective than RTSS. And we also need different types of vaccines that can not only prevent clinical disease and death in children, but thinking about elimination and eradication, we want to use vaccines as a mechanism for blocking the transmission of parasite from one human to mosquitoes and then back to more human beings. So I'm going to dig a little bit into the science, not get into too much detail, but just to highlight here on this side, this is the life cycle, and I really have to show you this to understand really what we do in terms of development of vaccines and to understand the disease a little bit. So as Gina mentioned, the infection begins when you get bitten by an infected mosquito, and then the sporozoites, that's the form of the parasite that comes in on the bite, migrates to the liver where it infects the liver cells, and then over the course of about 2 weeks you get a massive amount of reproduction of the parasite, and then those parasites get released from the liver and then infect blood cells. And this is the part where you start to get sick. Okay, so for the first 2 weeks it's really what we call asymptomatic, you're not sick, but towards here, this is where we start to get massive amounts of parasites, start to get anemia, and in severe cases may even lead to cerebral malaria or death. And then finally, the final stage in the life cycle in the human is that the mosquito then feeds on an infected human, takes up a different form of the parasite, and then over the course of a couple of weeks again, that mosquito becomes infectious to human beings, and they can go on and infect a different person. Okay, so very complex life cycle, and the parasite has really learned a way to evolve really superbly with both of its hosts. An important thing to note on this slide here is for many years, the drive was to try and reproduce the type of natural immunity that we see in the field. So if you get repeatedly exposed to the parasite, eventually you become immune, okay? You're protected and you don't get sick anymore. So for many years, the approach was to go after that type of immunity, which we call blood stage immunity. And what we've learned over the years of developing vaccines is that's a very challenging way to go for many reasons, and I'm not going to go into those right now. But the successes that we've really had to date have been targeting what we call the bottlenecks in the life cycle. The points in the life cycle where the parasite goes down to very small numbers, makes itself very scarce to try and evade the immune systems of the host. And that's really where we're having a lot of success. There's 3 main approaches to vaccine development. Trying to get it before it gets out of the liver, trying to target it when it's in the bloodstream, and then to try and prevent it being transmitted onto mosquitoes. We need different types of immunity. We need to use the vaccines to elicit different types of immune responses to target these different types of vaccines. And we also have different tools available to us to help develop these vaccines. We can't take everything that looks promising into Africa. It's absolutely not viable. So we have to use a series of down selection or prioritizing criteria that enable us to identify which are the most promising things to move into the field and to test in African field studies. And some of the models that we have, and one of the most exciting ones and useful ones that we've had over the years, is what we call the human challenge model. So that enables us to actually vaccinate people here in the US against an experimental vaccine and actually expose them to bites from infected mosquitoes. And over the years, that has really proven to be a very, very valuable and accurate tool in enabling us to predict how things will actually work when we take them into Africa. So we can go into Africa with things that we actually know have got a reasonably good chance of success. And Angela, I'm sure, you're going to talk more about that challenge model because we're actually supporting work here at Seattle BioMed to actually develop that model and actually use it for testing vaccines going forward. So it's really very exciting work. So where have we been successful? So Christian mentioned RTSS. This is a partnership with GSK. This is the vaccine sort of depicted here on the right, and this is a vaccine that protects about 50% of the time. So it's not 100% effective, but 50%, given the massive amount of disease burden from malaria, is still a tremendous achievement. And the other approach that's been very successful is if you take infected mosquitoes and then weaken the parasite by what we call irradiation, and have those mosquitoes feed on individuals, and after enough feeding rounds, people become immune to the parasite. Okay, so it's really taking the whole parasite approach. And Stefan's going to talk a little bit on some very novel approaches that his group is working on to develop vaccines that are based on this knowledge. And both of these vaccine approaches, very diverse, one using the whole parasite, one using just a small piece of it, were both shown to be effective using this challenge model that we use here in the US. Just before I finish, the final thing I want to talk about is a new area that we've really started to engage more and more in at MDI over the past couple of years, is what we call transmission blocking vaccine. Okay, so what does that mean? So most of the vaccines that we've worked on over the years for developing and implementing in Africa are really to prevent clinical disease, and death. But that's not always going to help us in our mission towards elimination and eradication. What we need to do is block transmission, block the cycle of transmission from human to mosquito and human to mosquito. So we have to target a different stage of the parasite to be successful in that area. So what we do is we look at a whole different range of targets. We look at the forms of the parasite that are actually infecting the mosquito, as opposed to those that are infecting the humans. And we've actually got a really remarkable model that we use to evaluate these types of vaccines. We can't test these vaccines directly in humans in the US because we don't have transmission, and we can't see if those vaccines work in that same manner. But we do have these fantastic tools, which this one's called the membrane feeding assay, which we actually can use in labs here in the US and show that a vaccine approach can actually protect mosquitoes from getting infected when they feed on infected blood. So again, we have tools that will enable us to down select and identify the most promising vaccine approaches that could have a role in elimination and eradication of malaria. Just as I finish, I just want to throw up our portfolio. These are the programs that we work on. As Christian mentioned, we rely on partnerships. We have a tremendous number of partnerships, and this really outlines those that we work on. So at the very end, the final stage of development, RTSS with GSK, and then earlier, a whole host of new programs that we're going to down select and move the most promising ones of those into the field studies in Africa over the coming years. So thank you very much.
Thank you.
Good evening, and thanks for coming. Since I'm a member of this institute, I'm delighted to have so many people being here tonight interested in malaria. As a researcher who doesn't get much out of the lab or the field, great to see that there's actually some public interest out there. Public interest is there. This is the recent cover of Science magazine focusing on TB and malaria. It's a great resource if you're interested in getting more detailed information about the current status of research and development to find interventions for these diseases. I refer you to this latest issue, will come out tomorrow. So we have heard about malaria, and we have heard about the problem, and we have heard about the basic strategies to prevent malaria or to treat malaria. We have heard about the numbers, but I think it is really important to remember that behind those numbers are individuals, and behind those numbers are children who get infected and suffer and die from malaria. So I just want to give you an example, which I still, as a scientist who has worked in malaria for 20 years, find incredible and hard to imagine. If you go to a region in Uganda or in the Congo, to a village where there are about 550 infectious bites per year, that means that a child gets exposed to the parasite, potentially lethal injection twice a day. Or imagine your child being exposed to such a disease. So we have to find a solution for this, no doubt about it. And I'm of the opinion that we need a vaccine to get rid of the disease. I personally think it's not possible to eradicate this disease with other means. I think it's important to control it and push it back and decrease the burden, but we need a vaccine to eliminate it. One of the big problems, obviously, with existing tools, as we heard, is that the mosquito becomes resistant to insecticides, the parasite becomes resistant to drugs. So you can see this as a kind of a never-ending battle and arms race between what we throw at these pathogens and at the vectors, and what they evolve to become basically resistant to those interventions. The fact is that still today 40% of the world's population is at risk of malaria. Malaria has been around since the old Egyptians, so I think we have to remember that this is a very difficult disease. So I talked briefly about vaccines. I actually introduced the topic already very nicely. I just want to point out there's really, for any type of vaccine, there's two basic strategies to develop a vaccine for a pathogen. One is the supplement approach, and the other one is the whole organism approach. The supplement vaccine approach is represented by the RTSS vaccine that actually described it, confers about 50% protection. And that is you just take a building block of the organism, of the infectious agent, produce it, immunize people, and hope that the immune response is diverse enough and good enough to protect against the incoming pathogen. The whole organism approach is you just take the whole pathogen, and obviously you can immunize people with lethal pathogens, so you have to weaken it. You have to what we call attenuate it to make it harmless, but still provides the immune system with a multitude of what we call antigens that allows the immune system to be trained to recognize the incoming pathogen and combat it. So if you use the whole pathogen, the immune system is really trained in an optimal way to detect and destroy the real thing, the infectious agent. With a subunit vaccine, the problem is it targets it on one particular aspect of this pathogen. And that can be dangerous because the pathogen can escape from this immune response relatively easily. So because of that, I think most of our successful vaccines still up to today are complete organism vaccines, what we call whole organism vaccines. And you see the most successful ones listed here, measles, mumps, rubella, polio. You know that smallpox has been eradicated with a vaccine, and there's actually another disease which is not human, the second infectious disease to be eradicated with a vaccine, and that's a disease called the Melinda pest. It's a disease of cattle in Africa, and has had tremendous negative economic impact on the African continent. And that's the second disease to go based on a vaccine. As we talked about the life cycle, here's a reason really why we don't have a vaccine. Very complex parasite, 6,000 genes, different forms, shapeshifter, constantly changes its makeup, escapes the immune response, is transmitted by mosquitoes, invades the body in very low numbers, and then amplifies to tremendous numbers and kills the host. So the subunit vaccine approach is really problematic in that we have so many potential targets to choose from. Here's a gene map of the parasite. You don't have to see the details. This is all the genes in the parasite, more than 5,000. Which one to choose from? That's a real challenge, I would say. And there are programs to identify new target antigens, and they are supported by the Malaria Vaccine Initiative. Our approach is slightly different. We really use the whole parasite, and here's the sporozoite stage that Ashley talked about. So what we try to do is we really try to take this whole form of the parasite and make it into a vaccine. But in order to accomplish this, we need to make it harmless. So it cannot cause disease when we vaccinate individuals with it. And the way we do this, we select genes that are critical for the parasite to initiate the infection in the liver. And I'm not going to go into details of how we found those genes, but of the 5,000 genes that are out there, there's about 30 or 40 that are critical for the parasite when it affects the liver to grow and cause and develop into disease-causing forms. And the nice thing about this technology is that you can actually track this vaccine very easily by genetic fingerprinting. So once you have this attenuated vaccine strain, you can always identify it. So it's always different from the real parasite, and by a simple genetic method, you can identify your vaccine strain, which we call genetic fingerprint. So we use these attenuated strains, and we obviously need to test if they are properly weakened, and that's a big challenge for human parasites because we don't have any animal models for these parasites. So what we use is mice that have human livers, and that sounds crazy, but it actually exists. And here you see a mouse with human hepatocytes, with human liver cells that is infected on the left with the wild-type parasites, these little green dots that you see in the center. And that parasite grows over a couple of days, and you can see after 4 days it becomes a big green blob. And that parasite is going to spawn a lot of new infectious forms. On the right side of the panel, you can see our vaccine strain. It affects the liver, it makes a little green dot, but it can't grow after 4 days, and it disappears from the liver. So the parasite's vaccine is attenuated and is not persistent in the liver. So it infects, but then disappears. But we do hope that this primes the immune response in a way that protects against subsequent infection. And we have tested this in animal models in mice, and this is just a simple vaccination experiment that I show you here, where you can see when we immunize with this vaccine, we get complete protection against infectious parasite challenge. So this vaccine protects 100% against the lethal form of the parasite. And obviously we want to test this in humans, and this is the way we do it, and Angela Talley is going to talk more about this. But we vaccinate individuals with this vaccine candidate, and these are volunteers here in the United States, and then we challenge them with the infectious form of the parasite and test and ask the question, does that vaccine protect? And I can tell you that our first vaccine candidate based on this principle is actually in clinical studies as we speak. We started immunizations yesterday. So I'm handing it over to Angela, who will tell you a little bit more about how we test vaccines.
I'm very excited to tell you about a new development in partnership with M.P.H that we embarked on 2 years ago. I think you're all aware that for decades, Seattle BioMed has been a major global health research center and focused on discovery science. And over the last 2 years, we've made it a really exciting transition into moving those discoveries from bench to bedside. In partnership with M.P.H, we worked to establish the human challenge model at SBRI. So we set up the human challenge model at the Malaria Clinical Trial Center to be a global vaccine testing center to serve the expanding portfolio of malaria vaccine candidates coming down the pipeline over the next few years. By Seattle BioMed and its partners. And by leveraging the significant institute expertise, we're able to bring those discoveries from bench to bedside and accelerate the transition of only the most viable candidates into the field. So we are only the 4th center in the world capable of conducting the human challenge model, which is a critical testing tool to down select the most viable vaccine candidates. It's the standard model of testing malaria vaccine in safety and efficacy in early phase clinical trials for the last several decades. There have been numerous challenge trials conducted since the 1970s, and the vaccine that Ashley mentioned that's in phase 3 studies throughout Africa now started in human challenge studies 20 years ago. So it is extremely, lots of experience. We have a lot of experience in conducting this model. It's extremely safe and has a very simple design and simple readout for evaluating malaria vaccine efficacy, because unlike HIV vaccines, we can challenge people with malaria who've been vaccinated and see whether they get malaria. We can treat them right away and cure them right away. So participants are closely monitored and treated upon the first sign or symptom of malaria based on a positive blood smear, and they quickly recover. So it's an extremely safe model that allows us to address a variety of vaccine candidates in our expanding worldwide portfolio. So the parasites are grown in culture in our controlled facility upstairs, and the mosquitoes are infected by the membrane feeding method that Ashley pointed out earlier. And as Stefan was demonstrating on himself for Bill Gates, the participants are infected by the bite of infected mosquitoes by putting their arm over a cup, and the mosquitoes are allowed to feed on the participant arm for 5 minutes, after which the mosquitoes are dissected to evaluate that they had sufficient numbers of parasites going into the participants. So here in Seattle, we do our clinical evaluations here at Seattle BioMed, where we also grow the infected mosquitoes in the secure challenge facility. There are 8 levels of containment for these infected mosquitoes, so that there's not really any chance that they would ever see Westlake Avenue. And we do the human challenge infections here also, and then participants around the time that they're expected to become parasitemic or develop clinical signs and symptoms, they go into a local hotel where we move the entire clinical operations over there with all of our study staff and monitor the participants very closely every single day, and we have staff on there on site, so that if anything so much as a hangnail develops in these participants, we are assessing them for malaria right away, and many of the participants are diagnosed before they ever develop symptoms, and so that we are treating it much sooner than if you had developed malaria out on safari and gotten sick and gone to the hospital. So here at Seattle BioMed, the Malaria Clinical Trial Center actually comprises multiple components, including the clinical component, which is our clinical facility. It's up on the 4th floor. For those of you who are going on a tour later, you'll see that. We also have the SATMIR, the secure insectary, where we grow the infected materials, and the participants are challenged. We also have the clinical translational research laboratory on the 2nd floor, which supports the immunology assessment and the immune assays and tests using the trial samples and using the blood samples coming out of the participants from the trial. So the path to developing this challenge facility has been long and somewhat complicated. There's a very complicated regulatory environment, as you would hope, for something that sounds kind of scary. So the FDA has reviewed every single step of everything that we are doing. And we have a very extensive and lengthy development process in each and every trial that we conduct here. And each and every trial is reviewed by an ethical review committee. And once we get sign-off from them, we conduct it per standardized procedures. And we are really excited to tell you that our last study visit of our very first trial is next week. And we have, at this point, successfully demonstrated the successful implementation of the human challenge model at SBRI. And as of the end of March, we successfully infected 6 people with malaria, demonstrating that our model is working. And our participants were extremely enthusiastic and extremely global health oriented. They were very excited to know that they were going down in Seattle BioMed history and Seattle history. And this is just the recruitment campaign that got them here, but I think it points out a really important component that a child dies every 30 seconds of malaria, and there's not that much needed for people, every person in this room to help. So stay tuned as we move forward, taking our discoveries from bench to bedside.
Thank you very much. So let's go ahead and open this to questions. Yes, please, up here.
How did you recruit your participants, and are you going to take care of them forever if the vaccine doesn't work?
So that's a good question. How did you recruit the participants, and are you going to take care of them forever if the vaccine didn't work?
So the benefit that we have establishing the Malaria Clinical Trial Center here in Seattle.
You need to put the microphone right up to you. There you go.
So we benefit from having a significant global health activist community in Seattle, and people here are very motivated to come out and do what they can to support the cause of developing malaria vaccine. So we did a very limited recruitment campaign, and by word of mouth and some of the advertising that you saw, we managed to get 250 people contacting the site within a few weeks, looking to volunteer for a study where we were only enrolling 6 people. So, and similarly on previous efforts, we in an early press release, we received over 2,000 contacts to Seattle BioMed for people who are wanting to participate. And to your second question, the participants are entirely covered for all of their any sort of medical injury that might happen to them, although I do have to highlight the fact that this model has been conducted by our partners who helped us stand up the facility at Walter Reed, and they've conducted the human challenge model in over 900 people to date, and never once has anybody been hospitalized.
I think the difference is this is a treatable disease, so you're treated and then you're done.
You're being treated, right?
No, that was years ago. I'm healthy. Yes, no, it was falciparum, so it's not a chronic disease, it's an acute disease. Yes,
The
question?
mosquitoes that infect people, do they do any good? And is it feasible to wipe out the mosquitoes?
That's an excellent question. Who would like to take that one? There are people who are working on that. Although mosquitoes do some things like they feed birds. And so I don't necessarily want to wipe out the birds that depend on mosquitoes. But there are approaches towards either getting rid of the mosquitoes or making them have a lower reproductive rate so that they can continue to expand. The interesting thing is that very few mosquitoes, even in high-intensity areas, are infected. Maybe 1% of mosquitoes. So it actually takes a really interesting dynamic and dynamic approach to make that approach work. Now that's not a vaccine, but you're right. It would prevent the transfer of the disease from one person to another from the mosquito. So it is via the mosquito. So it is a viable, intellectual, and scientific approach. But I don't know what the data is. It's still an evolution. Don't have the answer for you.
Isn't the good better than what you're going to lose?
Don't have to answer that right now. The first answer is, does it work? Then we can answer the merits of it. Other questions? Yes, thank you.
I have two questions. Number one, why do African mosquitoes transmit malaria and our mosquitoes don't? But secondly, have you looked at the fact that some people are more susceptible to mosquito bites than others? I go camping and hardly get any people with me get filled with mosquito bites.
Yes, some people are delicious and some people are not. That's true. Would anyone like to tackle that, Stefan?
Well, you have mosquitoes in the United States that are perfectly capable of transmitting malaria, number one. So we have actually quite frequently local outbreaks of malaria, especially around airports. I remember vividly when I was at New York University School of Medicine, we had a malaria outbreak on Long Island. An individual infected with malaria didn't get treated, went on a weekend trip to Long Island, got bitten by local mosquitoes and transmitted it to about 20 Boy Scouts out in the woods camping. So you have mosquitoes in the US that can transmit malaria. You just don't have the malaria parasite around on a regular basis. So it's not really a stable transmission situation. I want to also remind you that up to the 50s there was regular malaria transmission in the United States, especially in the southern part of the United States. Alabama, Mississippi, Louisiana, malaria was the #1 disease in the last century. So it's not been gone from the United States for a long time. We managed to get rid of it in the 50s with many different interventions. The most important one being TB, because people started to stay inside and not hang out on the porch anymore. So with regard to your question, so there's many different mosquito species, obviously, and you may be very tasty to a mosquito here in Seattle and you're not tasty to a mosquito in Africa or vice versa. Every mosquito species has different preferences. Like, well, you have different preferences than the person next to you, so different mosquito species have also different preferences, different smells, different tastes, and so on and so forth. It is actually a very interesting science, and people try to build repellents, mosquito repellents, based on the smell and the taste of the mosquito. The most famous one being Limburger cheese, which is very attractive to mosquitoes and can actually be used as a mosquito trap. So, to answer your question, mosquitoes bite people at different frequencies, but in Africa, where transmission is highest, there's one species of mosquito called Anopheles gambiae. And this mosquito bites only humans. It doesn't, for instance, bite any animals like many other mosquito species. So it's very effective of bringing the parasite from one human to the next human. And that is what is making the transmission situation in Africa particularly problematic.
All right, we have a question back here. Yes, please, if you can speak up.
Is it known that the human's the only other mammalian in the reservoir for this? If you vaccinate the humans, does that solve the problem?
Good question.
Or are they pigs or cows or something?
Turns out each species has its own kind of malaria. Stefan?
Well, so there's many different malaria parasites, but they are mostly specific for a certain species. So you have malaria parasites in birds, you have malaria parasites in rats, you have malaria parasites in monkeys. But at least until recently, we thought that they're staying within their particular host. But what we learned over the last couple of years is that actually monkey parasites, especially in Asia, can be transmitted to humans and can infect humans. So we have a big problem there because it's not only a human to human transmission situation is also a transmission situation where parasites can be transmitted from non-human primates, from monkeys to humans. And lastly, it was recently found that especially in Africa, in great apes and chimpanzees, there's a lot of parasites in these apes that can be transmitted to humans. And once they establish an infection in the human, they can actually be transmitted from human to human. So it's not as bad as with other diseases in terms of animal reservoirs, but there is a problem there, especially in Africa and Asia.
We had another question further up here? Yes.
So my question is
There's a microphone for you, sir.
about the expectation of the price of the vaccine when hopefully it will be approved for use. And is there any plans or expectation how it will be procured or provided for those, as you said, poor people in Africa?
So let's ask both Christian and Stefan to talk about what they think about of their respective vaccines, so RTSS and the genetically modified. Who would manufacture it? Because that's one question. Someone's got to make it for this low market potential. And then what do we know so far about the price? Stefan, why don't you start with RTSS?
As far as the RTSS is concerned, we cannot, as the plant has been built already, we cannot start a phase 3 without having the commercial facility being built for a minimum capacity that will ensure that the vaccine is going to be available if licensed and successful across that phase. In January, there was a meeting in New York during which Andrew Vitti, the chairman of the CEO of GSK, made a very interesting commitment saying that the vaccine would be made available at cost of manufacture plus 5%, and that's on record. So that's something which is a major commitment from our partner to ensure that the vaccine is going to be at a very reasonable cost. So the next part of your question, how much is that going to cost to the mother in Africa? Nothing. There are systems at the moment that are in place to ensure, Gavi, for example, systems that are in place to ensure that the funds are available for the vaccine for those countries.
What do you know about what is your thinking to date about the whole parasite approach?
I think right now we don't really have a robust industrial production platform for a live attenuated malaria vaccine, although it seems to be the most promising in terms of efficacy. What we know about it so far, it's 100% efficacious or 98% efficacious based on historic data. But it's obviously very difficult to manufacture, so I cannot tell you really what in the end the vaccine will cost. There's a lot of work to be done in terms of establishing robust manufacturing platforms for such a vaccine and the formulation and so on and so forth. But I just want to echo what Christian has been saying. For the mother in Africa, the goal is the vaccine will not cost anything. Because someone who makes $2 a day will not be able to afford a vaccine that costs more than $200. So I think it will be distributed for free, like the bed nets are distributed right now.
So another thing to think about is what makes up the cost of a vaccine. And it has certain components. One is what it costs to actually manufacture it. And that, will depend on many things that still need to be worked out. The other part is the investments that have to be made during the development process. And for a malaria vaccine, it's coming from places like NIH and foundations like the Bill and Melinda Gates Foundation. They are funding in partnership, offsetting some of the costs of industry, but a great deal of what would otherwise be required by industry. And I think that the third part is profit. And let me tell you, no one who's working in malaria vaccines is thinking they're going to make an enormous amount of profit. It's not the driver for why industry or biotech or the not-for-profit sector is engaging. It's those million children that are dying every year. Question up front?
The last time I was here for your malaria vaccine, I think it was you, Stefan, that talked about just what you're learning in terms of developing a vaccine for a parasite because it's never been done before. So based on just like basic science, what are you learning that will be applicable in the scientific field in general that we may have never known had we not gone down this path?
So Stefan, if you could repeat the question, please, so that the people in the back can hear.
Well, the question was, besides the fact that we want to make a vaccine that protects people, what do we actually learn from these early stage testing trials in humans? Is that correct? Well, I think we learn a lot about the immune system and how it responds to vaccination against parasitic disease. Right now, we do not understand how our immune system actually responds to the parasite. There's many studies in the field to look at natural infection, but this gets very complicated because the kid in Africa is repeatedly exposed again and again and again to the parasite. It has parasites in its bloodstream for a long time, so the immune system reacts in a very complex way. So these human studies, these early human studies here in the United States allow us to ask questions in people that have never seen malaria before. And then we can actually dissect the immune response and understand what part of the immune response is critical to protect. So our vaccines are kind of probes or tools that make us understand what arm of the immune response is critical to protect. For our particular type of vaccine, I want to emphasize that it's very important to determine the safety of these vaccines, because the problem with live attenuated vaccines of any type, you can think about influenza, or you can think about even polio, that they are the real thing and they might cause infection. Or, you know, in polio, with a live attenuated polio vaccine, 1 in 100,000 kids.
1 in a million.
1 in a million. 1 in a million. Thanks for correcting me. May actually get the disease. So it's always a cost-benefit analysis, obviously. But we want these vaccines to be as safe as possible. So again, these human early tests are very critical to determine safety for a live attenuated vaccine. We do not have this problem with the recombinant vaccines, the supplement vaccines for instance, the RTS vaccine that is in phase 3 studies right now, but because that cannot infect.
Let's take the question from the back, Melinda.
So Stefan, I noticed that with your vaccine, you still are using mosquitoes to give people the vaccine. So clearly that's not going to be feasible across Africa. It's a little hard to imagine the kids lined up with the cup on their arms. So what are your plans for, when do you transition this over into kind of becoming more like a real product, and how you would deliver it? And not at all to criticize what you're doing now, it's just to establish that it even works. And then to put the time and energy into it. But I'm just wondering, in the context of Seattle BioMed, how do you guys think about making that transition from, you've transitioned from basic research into now clinical research, then that transition to making real products? How are you guys thinking about that?
So this was a complex question, and I'm always glad to get these rather challenging questions, because it makes me think about these issues as well once in a while. First of all, yes, right now in these early clinical studies, we deliver the vaccine by mosquito bite. The reason we do this is because we do not know right now how to manufacture it outside the mosquito, and we would have to invest a lot of money to get it out of the mosquito and formulate it and vial it for injection. But for early clinical testing, that's completely fine, because we want to test early safety and efficacy, so mosquito delivery is perfectly fine. I'm particularly interested in actually starting to work on production methods that allow us to make the vaccine outside mosquitoes. And I think that's possible. It's a scientific challenge right now. And so people like me need to take it up and solve the problem how we can actually grow the parasite outside the mosquito. And I think we can do it. It just requires a lot of smart people and smart thinking and some investment to do it. And once we have a production platform that is, for instance, in tissue culture, like many other vaccines are produced in tissue culture, then we have to think about how to formulate it and how to vial it. And obviously for these steps, we need to partner with other organizations. We are not a developmental, we are not that far down the developmental path that we would be able to tackle these problems on our own. We are still working early in discovery and early clinical development, but for the later phases of manufacturing and development, we have to partner with other organizations. But I think it's critical, again, to re-emphasize this. It's critical that we have a tissue culture-based manufacturing platform to a live attenuated malaria vaccine.
So I have somewhat of a related question. So in a real field setting where the where the parasite is going through multiple cycles of infection, my understanding is that there's a high rate of mutation, which is one of the reasons why vaccination has been a problem. So in that scenario, how does the efficacy you determined by your human pathogen method really translate into a field setting? Because obviously in the lab that's not going through the same kind of cycling?
I think it's for your vaccine.
Well, yeah, I can take it.
Ashley, you take it.
So yeah, it's a good question, and we're limited in the challenge studies that we do here in the States. Is that it's very difficult to maintain a whole plethora of different parasites to be able to use in the challenge model. Okay, so most of the studies that have been done over the sort of last 20 or 30 years, these challenges are all using the same strain of the parasite, and oftentimes the vaccines are actually made to perfectly match that parasite. Okay, so we're biasing everything in the favor of success, and still, most of the time, things don't work. With RTSS, the first indication that we really got that the vaccine was able to confer protection in an environment, a real-world environment where you get diverse challenges, was actually in the field. So typically, you have to go to the field to demonstrate not only that the vaccine is working in the field, but also that it can protect against the diverse natural isolates. There is some work going on to actually expand a little bit on the number of strains that we have available to us to test in the US. And so there is some work ongoing that we're partially supporting that's going to open up 2 or 3 additional strains. And we look at very diverse strains across very diverse regions of Africa, so that going forward, maybe in 4 or 5 years' time, we could actually have even better information about the breadth of protection before we even go into the field.
All right, go ahead.
So I have a question about moderate levels of protection. Given the human model you're using for testing the attenuated live vaccine, how do you check for the possible existence of a level of, say, reduced symptom in a patient?
So in terms of recruiting the participants, we have an extensive list of exclusion criteria. And one of those a couple of those exclusion criteria that are key to the answer to your question is that we exclude people who have a recent malaria travel to a malaria endemic region, or anybody who's ever had diagnosed malaria in the past, or anybody who's taken antimalarials in the last few months.
So my question is more along the lines of, if your vaccine is capable of eliciting some protection but not completely eliminating clinical symptoms, but you're treating them so promptly when they have a blood smear, how do you find that sort of phenomenon?
So do you wait for clinical symptoms to treat, or are you screening through systematic blood drawing, looking for parasites? That's an example. How are you monitoring the patient?
Yeah, so we are monitoring the participants daily with daily blood smears. So we treat based on the positive blood smear.
You're not waiting for them to become ill.
Right.
The first sign of a parasite that gets treated.
So maybe just to follow up on this question, because I think you were going in a certain direction. How do you test for partial effectiveness or for effectiveness against disease? And so with this challenge, well, that's not possible right now. The only test that you can conduct is, does that vaccine protect against infection? And as soon as an individual is not protected, it is infected, you treat. So you have no idea if this vaccine has any impact on disease.
You've talked about protecting children, which is, I think, a great thing. I'm just curious to find out what your plans are for pregnant women who are also quite susceptible to malaria infection.
Is anyone working on that at this time at the table?
No.
There clearly have been considerations of developing a vaccine for pregnancy malaria. It takes a different set of targets is what's being evaluated for that. It's helpful to know that that intermittent presumptive therapy with drugs actually does help prevent disease in pregnant women. So we're not dependent just on a vaccine, that bednets does help prevent and lower the rate of infection in pregnant women. So it's like children, they're at higher risk for some unique reasons related to pregnancy, but we do have some ways of preventing disease for any kind of infection, not just malaria. Developing a product, particularly a vaccine targeted to pregnant women, is enormously challenging, not only in terms of ethics, but regulatory and market. The whole thing is very, very challenging. So it's a question whether that would be the first vaccine, in the first place that you would go with a vaccine, or if that's something that you would evaluate then an effective vaccine later on. And I suspect it may end up being a subsequent kind of study. a question up front.
My understanding is that MBI has shared information as the various candidates have progressed through the portfolio. Is that so, and how does that work?
Shared information with whom, if I could clarify your question?
Among the various developers of vaccines. I mean, are you sharing out the discoveries as you go along? Are you talking with one another?
If I could just comment from the foundation point of view, all our grantees are required to publish information. And what we really want to do, it doesn't really matter if you learn something that's negative data or actually even positive data. If it can't be shared rapidly with the community, with enough time to protect intellectual property concerns and to get it into peer reviews so that it actually does get published. But the thing is, it can't languish in the laboratory. It has to be published. And so they're hostage to that. They're hostage to us in that regard. Maybe other ways too. And then they pass on those requirements to their partners. So I think that's one thing. Anything else?
No, I think that's very much aligned with the strategy of Path MPI.
Let me ask Ashley or any of the participants another question. We've been talking, we didn't get into the species issues, but we've been talking about falciparum malaria. And the reason we've been doing that is because that's predominantly the one that kills. Vivax malaria is the one that becomes a chronic illness, which becomes much harder to clear because it hides in the liver. So what is the likelihood that a vaccine for falciparum malaria is going to protect against Vivax malaria? Would anyone like to answer that? I would say they'd be easy.
I'll try. Well, I think certainly with a very specialized, focused vaccine such as RTSS, which is really, as Stefan mentioned, is just taking a fraction of a percentage of the total parasite as part of the vaccine. There's very little shared regions between falciparum and Vivax, so we would not expect that type of vaccine to confer any sort of cross protection. I'll pass over to Stefan in a second, but just to highlight a really interesting development in the development of Vivax vaccines, which has really been a neglected area of vaccine development for malaria for many years. I think in large part because we don't see the large mortality with that disease that we do with falciparum. But recently, a challenge model has been developed at Walter Reed in Maryland to actually start evaluating Vivax vaccines in the challenge model in the US. And I think as we stressed through some of our presentations, having availability to that challenge model now for Vivax, we think is really going to help accelerate the development of Vivax vaccines as well, which is critically important. I don't know if Stefan has any comments relative to cross-protection when you're looking at more complex vaccines, such as whole attenuated parasites.
Well, falciparum and Vivax are what is different as humans and mosquitoes genetically. So it's a huge difference. These parasites have diverged in the course of evolution a long time ago. So indeed, as Ashley pointed out, it's very hard to get cross-protection because these parasites are so diverged. I think the likelihood to get considerable cross-protection is higher if you use the whole parasite as a vaccine because you have numerous targets and some of them may be very conserved. Some of the more important stuff in the parasite is highly conserved because the parasite has no function, and so maybe you get cross-protection. There's very little, but some scientific evidence that it actually does occur, that there's some cross-protection. In the end, I think we need to target Vivax with a Vivax-specific vaccine. I don't think a falciparum vaccine will be highly efficacious against Vivax malaria. I just want to point out one thing, and that's Vivax was always considered the benign, the nice parasite, and it's not a nice parasite. It's a nasty parasite, and it can make you terribly sick, and it does kill people. So I think we need to address that parasite as well.
Do we have a question in the back? Yes? Who's got the microphone? You control the world here.
Question about practicality of running a clinical trial in the field in say, Africa. That and local trust. Do you run into problems with governments not trusting what you're doing and local cultural taboos against trusting? I mean, the example of polio was almost eradicated. If I understand correctly, in sub-Saharan Africa, there was some mistrust about what the trials were doing or what the vaccinations were doing. Do you run into that kind of mistrust still? Is it a problem for doing these?
So for the clinical trial, the RTSS trial, we need to know that we have, at this point in time, we've got 40 ethical committees across, in fact, this trial in the US, in Africa, at the centers. prior to starting the trial, we always undergo a process of regulatory authorization to undertake this trial with the national regulatory authorities in the countries. The clinical centers are usually well-established centers that have a strong connection with the communities where they are working, and usually where they are conducting research in terms of understanding the perception of the development that they are doing. At the end of each and every phase of the trial, at the end of the phase 2, for example, the previous phase, the results of the trial were communicated to the communities for them to get a direct access to what came out of the trial, and it's always a detailed explanation of what's going to come for the next phase. At this point in time, with the development of malaria vaccine, we are also conducting research in terms of better understanding the perception of a vaccine and the perception of malaria. They're going to be very important data for us when we are going to move towards, in fact, the final stages of phase 3 and when we are going to plan the launch of that vaccine. For the time being, we have not yet faced the problem that polio did face in Nigeria, for example.
Let me add a comment to that. Clearly, concerns about vaccine safety, because you're creating an immune response, and that can be a reactogenic process, is a potential factor with any vaccine. And we are seeing concerns in the UK and the United States, as well as Nigeria, so it's not unique to developing countries. One thing that I found very interesting and helpful in visiting the clinical trial sites where these kinds of studies are being conducted is the care with which it's not just informed consent to the individual or to the family, but to the village and the village chief, so that the community is engaged. And one example is that in many places, there's a lot of fear about blood drawing. What does it mean to have my blood taken something else, and what are they doing with that blood? So that site was bringing in the village chiefs, and each village is, can be anything from a small group of households to a larger group of households. But they had a lot of villages, and they would bring in the village chiefs, and they'd take them to the lab, and they'd show how less than 1 cc of blood would be diluted in broth and then become a much larger looking specimen, and then how it was cultured and what they were doing with the cultures and what they would learn, and then how they would dispose of it. So they took them through the whole thing so they could answer what happens to my baby's blood. And it's that kind of engagement of the community and information about what the different aspects of the trial are that helps manage even through the clinical trial process. Let me close this session by asking one question, and each of the speakers to respond, and then I'm sure that they would be delighted to answer questions individually. You can come up afterwards. All of you are tackling a very different part of this whole R&D enterprise. And each piece requires, the public global health village, the issue of partnerships to make it possible. Could you talk briefly about what a single factor makes about a partnership makes it either feasible or challenging to get your job done? Just choose one. I'll pass it down. You're first.
That's an interesting one. I must say, based on the partnership that we have, it does vary a lot. Time is a very important factor, and over a period of time it's a very important factor in the evaluation of the partnership that we are going to start. I would say there is science, a certain number of elements that we are going to estimate during, in fact, the negotiation with a partner, but during that discussion we are also going to estimate the quality of the relationship with a partner, and I think that's a very important element to evaluate prior to signing the contract and embarking.
How about at the scientific level, where you're negotiating with companies?
I would say one of the big challenges is particularly the companies that we work with, the technologies that they're using to apply to malaria vaccine development, they actually are using to apply to very lucrative product opportunities as well. And so that requires that we have to walk a very fine line relative to working with those partners to ensure that there isn't any sort of negative impact on any data that might come out of the study that could impact a very profitable product for them. So I would say that that can be one of the challenges with the technologies are not just being used for malaria, they may also be used for cancer, HIV, TB vaccines. So I think that's one of the challenges we have.
Stefan?
Well, as a basic researcher, I think the challenge is always to understand when you have to move forward and actually face the real world and test something. But very few of us have actually the opportunity to test things. And I think the Grand Challenge in Global Health program was a very unique program because it allowed scientists to put the whole team together, what Ken liked to call the dream teams, and tackle the problem all the way from the basic discovery to potentially clinical testing. And I think that's a great model, and I think it's a very successful model. It requires large investments, large grants at a given time that fund everything from the basic discovery in the lab all the way to clinical testing. And that's a very effective process if the money is there to, from the beginning, to provide funding for all of these steps. Obviously not phase 3 testing because it costs hundreds of millions of dollars. So I think that's a very nice model, and I've enjoyed this model, fortunately, because I was one of the recipients of the grants. I think this program needs to be continued because there's so many other challenges, and I think we need this again. We need another Grand Challenge program. Because otherwise you do the basic research and then it's the end of the story, and many good ideas never get out of the lab, never get into this early clinical testing. And I think we need to do this more frequently, and we have great models for malaria, the clinical models, the challenge models, where we can test interventions very early, and we just need to do it more.
Okay, Angela, uniquely your partnerships are with the community.
Right, so as a clinical researcher, my partnership is with the people that I'm reaching out to with our messaging, and I think the challenge there is trying to convince people that a small amount of their time can make a huge impact, in history and moving, and for the people, for the millions of kids who are dying over the course of the next few years. So I think that people respond very well, at least in the Seattle community, to the global health messaging, and there's a strong activist component to that, but it's not the case nationwide, and we constantly have to reinforce the reason and rationale behind the science and make that link between the discovery science and moving it out to the field. And I think that's part education. I think it's part science and it's part medicine, and it's a really unique situation to be in. But I think that given the right approach, people respond very well, and we see that response on our doorstep over the last few months.
Great, thank you. Well, I don't have an answer to what happens next with Grand Challenges, but there is a program that's interesting that I'm sure everyone here knows about, but it bears repeating. One of the follow-up programs is called Grand Challenges Explorations, and it's kind of like a baby Grand Challenges, but it's looking for great ideas. And this is a program that has a commitment of $100 million, and they're doing it $100,000 at a time. There are calls for proposals twice a year. I think they're due next week, right? Calling, including usually for infectious diseases and often malaria. 2-page proposal, one great idea, $100,000 for 1 year to test the concept, and then the possibility of more robust grants following on. And I talk about this everywhere I go because it doesn't have to be a huge idea. It doesn't have to be the huge collaborations. It has to be a testable idea and a thought. And what's interesting out of the almost 300 grants that have been funded so far is we've got a Nobel laureate in there, we've got a postdoc, and I'm actually talking to college students because they don't have boxes around their ideas. They're not inhibited by thinking, I'm trying to get them, to get the colleges to get them to apply as part of their homework, because I actually think there's a lot of great ideas out there, and clearly what we need in global health is innovation. Thank you, Seattle BioMed, for hosting this, and let me turn this back to you to guide them to the tours.
I'd like to ask you to please yet again thank the panelists and the moderator for what was a fantastic evening. So. I'd can tell you that I, for one, feel privileged to be working with these people. I feel excited and just very fortunate to be here at this time working on such an important problem. And I'm really glad that you're here to learn about what we're doing here. And when I say we, I don't mean just us in the building, we as this community. One of the values that we have at the Institute is learning. It's over there on our poster that talks about our mission, vision, and values. And I want to thank you for coming here tonight to learn more about what is not only a really important and challenging problem, but also to learn about the hope and learn about the work that's being done to address and deal with this problem.